As the body ages, a slow but relentless transformation unfolds at the level of the genome, not through mutations, but through epigenetic changes that reshape gene expression without altering DNA sequence. This process, known as epigenetic drift, is driven by cumulative environmental exposures, oxidative stress, metabolic shifts, and cellular wear. It involves changes in DNA methylation, histone modification, and chromatin remodeling that gradually deregulate critical biological pathways. Among the most affected systems is autophagy, the cell's internal recycling and quality control process. As autophagy-related genes become epigenetically silenced or misregulated with age, the cell loses its ability to maintain homeostasis, setting the stage for both tissue degeneration and increased vulnerability to cancer. Epigenetic drift is not a random process. Large-scale studies have shown that certain regions of the genome, particularly those involved in stress response, DNA repair, and metabolic regulation, undergo predictable changes in methylation patterns over time. Autophagy-related genes like BECN1, ATG5, LC3, and ULK1 are among those whose expression is altered with aging. For instance, hypermethylation of the BECN1 promoter reduces the cell's ability to initiate autophagy under stress. Similarly, repressive histone marks on ATG genes impair the formation of autophagosomes. The result is a chronic, systemic decline in autophagic activity, a slow erosion of the cell's cleanup crew. This decline comes with serious consequences. Without efficient autophagy, cells accumulate damaged mitochondria, misfolded proteins, and other toxic debris. In young cells, these elements are swiftly recycled or degraded. In older cells, they persist and cause further stress, triggering inflammation, genomic instability, and senescence. The very conditions that autophagy is designed to suppress become more prevalent, accelerating tissue aging and dysfunction. Critically, this breakdown in autophagy also intersects with cancer risk. Damaged mitochondria generate excessive reactive oxygen species, ROS, which cause DNA lesions and mutations. Impaired autophagy fails to clear premalignant cells or regulate oncogenic signaling pathways like mTOR and p53. Meanwhile, the epigenetic silencing of tumor suppressor genes and DNA repair enzymes, another hallmark of aging, further compromises genomic integrity. In this pro-inflammatory, damage-prone environment, cancer is more likely to emerge, and once it does, the impaired cellular surveillance systems of the aged body are less able to contain it. There is a dangerous irony here. Autophagy, which in youth acts as a tumor-suppressive force by eliminating damaged components and maintaining cellular fitness, is undermined over time by the very epigenetic shifts that accompany aging. Worse still, some of these epigenetic changes mimic those seen in early carcinogenesis. For example, BECN1 silencing is frequently observed in breast, ovarian, and prostate cancers. This raises the unsettling possibility that aging itself primes tissues for transformation, not through overt mutation alone, but through the quiet, gradual repression of protective pathways like autophagy. Yet amid these warnings lies a promising avenue for intervention. Epigenetic modifications are reversible. Unlike genetic mutations, they can be modified through dietary, pharmacological, and lifestyle strategies. Compounds like resveratrol, curcumin, and spermidine have shown potential in animal models to reverse age-related epigenetic silencing of autophagy genes. Histone deacetylase inhibitors, HDAC inhibitors, and DNA methyltransferase inhibitors, DNMT inhibitors, currently used in some cancer therapies, may one day be repurposed to restore autophagic competence in aging tissues, though such approaches require extreme caution, given their systemic effects. Furthermore, caloric restriction and intermittent fasting, both of which promote autophagy, appear to work in part by reshaping the epigenome in beneficial ways. These metabolic stressors activate transcription factors such as FOXO and PGC1A that enhance autophagy and mitochondrial biogenesis while preserving genomic stability. Importantly, they also counteract the drift toward global DNA hypermethylation that accompanies aging, maintaining more youthful patterns of gene expression. Still, a critical caveat must be kept in mind. Not all epigenetic changes with age are harmful, and attempts to reset the epigenome may carry unintended consequences. Inappropriate reactivation of suppressed genes, including those with oncogenic potential, could accelerate, rather than prevent, malignant transformation. Any therapy aimed at rejuvenating autophagy through epigenetic reprogramming must distinguish between protective and pathological silencing, a challenge that modern epigenetics has yet to fully resolve. In conclusion, the aging epigenome exerts a profound effect on autophagy, gradually silencing genes that protect cells from damage and degeneration. This suppression leaves cells vulnerable to dysfunction and cancer, not because of a single catastrophic event, but due to a slow, cumulative erosion of protective capacity. Yet this process is not set in stone. With careful intervention, through nutrition, pharmacology, and possibly targeted epigenetic modulation, it may be possible to restore autophagic function, extend health span, and reduce cancer risk in aging individuals. The challenge is not just to decode the language of the aging genome, but to speak it fluently enough to reverse its most dangerous messages without unleashing new threats in the process.